your nice introduction. Good morning, good afternoon, everybody. It's a great pleasure for me to be part of this conference and present some of our recent outputs. The title of my talk today is Understanding the Effects of Mutations on protein drug targets of pathogens for rational drug research and development. We are located in a small university town, Makanda Gramstan, in South Africa. And my group's research interest is in structural bioinformatics and the application of structural bioinformatics to disease-related questions. So, as we all know, structure-based computational drug discovery has successfully been used for the design of drugs for the treatment of both communicable and non-communicable diseases. We have many examples, drugs, FDA-approved drugs that is basically coming from computational drug discovery approaches. However, traditional structure-based computational drug discovery misses a number of important aspects currently, one of which is the consideration of the impact of evolutionary mutations of pathogens, including those linked to the drug resistance. So currently, this is mostly undetermined. And we believe that information gleaned may help to design alternative drugs that could potentially produce drug resistance effects to produce a longer lasting treatment options. So when we started looking at this problem, the problem of how to analyze missense mutations located in proteins in 2017, we realized that there's hardly any research on that, and there's no well-established pipeline or tool to do so. So my PhD student, David Brown, and I, that time, put a protocol together and published in this article. And this protocol was basically bringing all the known bits and pieces approaches together. But on top of that, we proposed that one should actually use molecular dynamics together with something called dynamic residue network analysis. So to that point, actually, residue network analysis or residue interaction network analysis, which was called RIN, over single conformation structures was already known. There were some analysis going on by using a crystal structure or minimized structure to build the networks of the proteins. But we that time identified that RIN applied to these static structures might not give consistent results, and it basically depends on how you are treating the structure. If you are minimizing, okay, so depending on how you are using this structure and calculating these networks, you could get inconsistent results. Thus, we propose something called dynamic residue network, and basically these calculations implement the concept of averaging the residue network interaction network metrics over MD simulations. Okay, so basically we carry it from the calculations from structures, static structures, molecular dynamic simulations, and then we actually that year published a tool, MD task. This is highly used tool to calculate dynamic residue networks, and more recently we established a web server. Okay, and this web server actually has many other tools, but it also has you can do DRN calculations as well as RIN calculations. So today, what I would like to show you is actually over 3 examples here, how important it is to combine molecular dynamics and the variations of network analysis to be able to understand mutation effects in protein structures. So the first example is from Hiv protease. This was one of our earliest studies done by that time my PhD student Olivier Sheikh Amamoudi. Okay, so we all know that HIV protease has lots of drug resistance mutations. Our research question here was, is there a common resistance mechanism against all protease inhibitors? What you are seeing here is the HIV protease structure, and these red dots are the drug specific resistant mutations. That time we looked at 8 drugs as listed here. Okay, these are drugs, and for each drug, we retrieved 100 highly resistant sequences and 100 hyper susceptible sequences. Okay, so 200 sequences per drug, and we build the structures with these mutations, which was totaling 1,600 structures, and we did the docking. We docked the relevant drug to the proteins and did MD simulations. These were short MD simulations repeated twice. And then we applied residue network analysis coupled with statistical tests. And this approach we call statistically guided network analysis, SGA. So what we saw from our results, regardless of behavior. So basically it was expanding laterally and pushing from down. And we hypothesized that actually these mutations, regardless where they
Ozlem?
are, yes,
I think we lost you the slides, so not sure how you can solve that. Should we
Yeah.
wait or?
Sorry, there's a bit of a hiccup and your screen share stopped. If you wouldn't mind clicking the stop share and then just reshare your slides, that should do it for us.
sure. Where is my stop share? Share. Okay, I'm resharing. I didn't stop anything.
Okay.
Okay, if it's working.
I can see your screen. Yep.
Okay, but
There we go.
okay. Sorry,
So
something
now,
happened.
sorry, that one stopped it. So now we need to reshare.
Okay. Okay. Do you see now?
I can see your screen. Yeah.
Yeah.
Perfect. Right. Thank you very much.
Okay. Okay, cool. So what we observed here was actually regardless of the drugs and a kind of common behavior of the protease. Basically, it was laterally expanding and a kind of pushing from the bottom part of the HIV protease, and we hypothesized that by this common behavior, actually the drug could be dislocated, and that would be the reason of the drug resistance. Okay, our second example is from TB about the drug pyrazinamide, PZA. I will call it PZA. It's much easier to say. Okay, so PZA is processed by the enzyme PZase, and we gathered 83 mutations here. Again, the mutations are all around the protein, not only in the active site, yet they are causing the resistance. So here we did something little bit different. We did long MDs and identified this in some of the cases actually drug was leaving the protein. We established an algorithm to analyze the unbinding events. So we cluster the proteins with the drug stays in the protein, and the ones leaving the protein. So two clusters, and again applied our statistically guided network analysis, And we saw that in certain mutations, again, the active site flap is opening, and it is the reason why the drug was unbinding. So, and regardless of these mutations, I mean, this is a common behavior that we observed in the second case. So, my third example is from SARS-CoV-2 AMPRO. As we all know that it's drug target, but actually this drug target has lots of mutations, and we did this study last year with the early evolutionary mutations. We got 50 mutant sequences from this site, and again, these mutations were all around the structure. The protein is homodimer. Okay, so after MD simulations, just even globally looking at the structures in RMSD level, we saw that some of the mutations were causing a different behavior than the reference protein that we got. Okay, and we found number of interesting results in this and published in the article. I'm gonna present only one of them, as this is a homodimer, and we looked at how these protomers are behaving in the presence of mutations, and we measured the center of mass distances between interprotomers. And as you can see, in the presence of some of the mutations, this distance was quite a bit of varying. And we also did dynamic residue network analysis to identify highly important residues, key residues, functional residues, and two of them were 17 and 128. And we have a further study on that, and we are still picking up these two residues as highly important, functionally important residues. So in conclusion, in a number of studies, we show that mutations might affect the structure, hence the function of the protein. These mutations, even though they look random, they are not randomly produced by the pathogens. Actually, they may lead a common behavior in the protein for drug resistance purposes. And these potential effects should be considered during computational drug discovery to design alternative drugs that could potentially reduce drug resistance effect. So last but not least, I would like to acknowledge CHPC, of course, for all our computational molecular dynamics simulations, and our funders. And also I would like to thank organizers, conference organizers, for inviting me to this nice conference. Thank you.
Wow. Thanks, Ozlem. That's a really interesting talk. I'm sure many of us are very curious to use these tools. I don't have questions in the Q&A tab, except that Peter is asking about the drug. What is the drug that had been used for the SARS-CoV-2 analysis?
Oh, no, there is no drug on that, because there is no drug. So in that, we only looked at the mutations to understand how the protein is behaving. So that information would be quite useful while designing the inhibitors. So that's
Okay.
what it was.
There's maybe there's a question now. So it's by Ruben Cloet. So he say, "Hi Ozlem, did you only look at the single or multiple mutation introduced in the structure?"
Single or multiple? Sorry.
Mutations introduced in the structure. So that can we use like multiple mutations to study the
Yeah, you can use as many as possible. I mean, so in MPRO analysis, some of the strains had multiple mutations.
Okay, and then this one question from me: can we use these tools as a standalone? Do you have a standalone versions for these tools, or are they only implemented as a web server?
No, MD task is in GitHub, and even MDM task, even though it's web server, the scripts are in GitHub, and we are hoping that people, if they use the scripts, they will just cite the MDM task. But everything is